All right. I hope everyone enjoyed the sessions this afternoon. I think we can all agree there were some really great talks. Hats off to the grad students and postdocs especially who presented some of their work to us this afternoon. So next we have a panel discussion to share some research that's been ongoing. Collectively, our world is focused on sustainability and the many aspects of sustainability that encompasses, and no doubt animal agriculture has a role to play in that. This has been something that's been a focus on campus increasingly and is now a part of several initiatives and has been a focus of researchers in the college for many years. So this is not new to any of us. But one thing that's been really exciting is that with innovation, the Dairy Innovation Hub funding for equipment and projects, we've been able to jumpstart a lot of research areas. And then as that research has gone on, been able to leverage those preliminary data for more external funding and have those research programs grow even larger. One of the keys that we set out to do in the beginning with capital equipment purchases, for example, was to remove bottlenecks or limitations. What are the greatest questions that we could answer if we had, and in some of those cases it was people, in some of those cases it was equipment, in some of those cases it was collaborative capacity, right? And so this is an example that you'll hear several of those themes throughout as we discuss. So we're going to feature this one, and it's my great pleasure to introduce some of my colleagues here as our panelists. So first, we have Francisco Peñagaricano down on the far end, who's an assistant professor, at least officially, until the turn of the academic year, who is in the Department of Animal and Dairy Science here at UW-Madison. And his research analyzes the genetic architecture and economically relevant traits in livestock. Francisco is the PI on the project that we'll be featuring for the panel discussion. And in just a moment, he'll explain a little more about that. Next, we have Lizzy French in the middle, who is a research animal scientist at USDA ARS Dairy Forage Research Center here in Madison. And I'd like to be the first to publicly note that she just found out that she is a senior scientist as of this morning, which is the equivalent of passing probationary period. Sorry, Lizzy. So Lizzy is alumni of our program in the department. In fact, she was on my floor doing research as a grad student as I started. So this is one of those full circle collaborations that here at UW-Madison we get a chance to experience so often. Okay. Lizzy's research focuses on using animal data to develop algorithms for designing rations and rumen microbiome modifications to enhance farm sustainability. And last, immediately to my right, we have Dr. Hilario Mantovani, who's an assistant professor, again, in the Department of Animal and Dairy Sciences here at UW-Madison. He specializes in rumen microbial physiology. And importantly, Hilario was a hub-funded faculty hire in our first round of faculty hires. So we're thrilled to have all three of them here with us today. So the focus of today's discussion will be the project funded under the Greener Cattle Initiative, and I'll hand it over to Francisco to give you a little bit of an overview about the project and what the efforts have been focused on.
Thanks, Heather, for the introduction, and thanks for the organizing committee for the invitation. Let me see if I know how this works. There you go. So given the importance of methane emissions, and if you think about it, there are multiple benefits associated with reducing methane emissions from dairy cattle, including reducing the environmental impact of dairy farming and also improving production efficiency, because beyond the impact on the environment, we can think of methane as an energy sink, as a loss of energy. We decided to address this problem using three complementary approaches, including selective breeding, genetic selection, the use of milk spectra data to predict methane emissions, and the manipulation of the rumen microbiome. And we are a group of multidisciplinary people, including geneticists, nutritionists, and microbiologists, from multiple institutions, including 4 land-grant universities, including the University of Wisconsin, Iowa State, the University of Florida, and Michigan State University, the USDA, and also the Council on Dairy Cattle Breeding, that is the entity, the organization in this country that runs the genetic evaluations. And again, our goal is basically address the mitigation of methane emission from dairy cattle breeding using 3 complementary approaches. So basically, there we go. Our plan is to phenotype mid-lactation Holstein cows. Our original plan was to phenotype or measure methane emissions and other traits in research farms, but we have expanded that plan and we are also working on commercial farms. And what we basically do is we measure methane emissions daily, feed intake daily, and milk production daily. And then we have milk composition roughly 4 times a week. And we have body weight every week or every 2 weeks. And basically, we will tackle basically 3 main objectives. One is selective breeding or genetic selection. Right now, if a farmer wants to breed cows for reducing methane emissions, we do not have the tools to do that. And our plan is to develop those tools so in the near future, farmers in this country, they can breed cows for reducing methane emissions. The second one is we want to develop a predictive tool in case that in the near future, either the federal government or the industry want to minimize methane emissions, the farmers, they need to identify what are the cows that are emitting more methane in their farm. And we are planning to develop a prediction tool using the spectra of the milk. And the third one, because 100% of the rumen, of the methane, is produced in the rumen, we need a better understanding of the microbiome in terms of composition of activity and how that microbiome is related with methane emissions. And for that, we will evaluate the rumen microbiome of cows with extreme methane emissions, very high, very low. And we are also planning to do some cool stuff where we will exchange the rumen between cows to try to decipher what are the relative contributions of the host, the cow, versus the microbiome when we talk about methane emissions. So in terms of solutions, this is a 3-year project. So first of all, we want to develop genomic solutions. That means we want to implement a national genetic evaluation for methane emission traits. And as I will show you later, maybe I'm biased, I'm a geneticist, but genetic selection is a very powerful tool because the changes that you achieve through genetic selection are permanent, cumulative, and incremental. So it's a very cost-effective and a very powerful tool for making changes in the population. The second one is what we call spectra-based solutions. If we can use the spectra of the milk to predict methane emissions, that is a win-win. Think about it. The spectra is, by definition, a very high throughput method, non-invasive at all, very cheap, that is used nationwide. So if we can develop predictions based on spectra data, then in the future, when farmers send their milk sample for testing, they will also receive not only composition, but also methane production of their cows in the farm. So they will be able to identify what are the cows that are emitting more, and maybe for those specific cows, use some specific interventions in terms of feed additives. And the third one is, after this project, we will better understand the rumen microbiome of these very high emitters versus low emitters cows. and we will have a better idea of what are the relative contributions of the host or the cow genome versus the rumen microbiome when we talk about methane emissions.
Great, thanks for that overview, Francisco. So as Francisco mentioned, this effort has been going on for even before our time, probably almost 15 years now, and historically has always included a geneticist and a nutritionist at each research station. And that's for a pretty clearly defined reason. The geneticists want to make genetic progress, but they have to have phenotypes for that. So there's some pretty long-running jokes that, of course, the geneticists can't do their jobs without the nutritionist. And we're the ones that actually do all the hard work collecting the phenotypes, right? So Lizzy, Francisco very casually said we're going to collect 4,000 phenotypes in 3 years, which is quite a big undertaking. Can you share with everyone what goes into that? Maybe what are some of the systems across the research stations that we use?
Yes, absolutely. And I think one of the powerful parts of this project is the continuity of data collection and how we're gathering the phenotypes. So on the slide behind here, you see a variety of methodologies utilized to collect enteric methane on ruminants. And they range from everything from the respiration chamber, which is limited in the number of animals, to those such as sniffers, which require more maintenance of the equipment and can be more drift in the measurements themselves. So what's unique about this project is that we're utilizing CLOC's green feed systems to collect spot samples on on methane, carbon dioxide, and oxygen. And across all the different systems, we have, again, these green feed systems. So as we look, for instance, at Arlington, we have the ability in various pens to have 64 measurements, or 64 cows that we're sampling methane on. And the pen 30 at Blaine Dairy is one example of that, in addition to pen number 4 at Arlington as well, where we're able to gather another 64 individual samples from 64 cows. And while we're taking this methane data, we're also collecting, as Francisco mentioned, body weight data, even body size and hip height data, milk production, milk composition. And so while we're making the majority of measurements on enteric methane at Arlington in a freestall setting, also, at Marshfield as well, we have an additional pen where we're doing freestall settings with green feed units. At the Dairy Forage Research Center, we have 150 tie stall spots where we are collecting methane measurements utilizing also the green feed system, as you can see here in a tie stall setting. And that's a little bit of a different scenario, whereas in the freestall, the green feeds are in the pen, the animals visit at their leisure, given a certain time allocation. But in this situation, in a tie stall, we're physically bringing the machine to each of the cows on the study as we go through the research project. The other major, phenotypic trait that we need to collect for this genetic improvement is intake data. And so as you can see behind me, there's a variety of systems, whether it's in Syntex that we have at Arlington, the Kalen doors that are at Marshfield, or again in a tie stall setting such as DFRC. And each of these examples, again, there's continuity across the other research institutions that are involved in this project. So being able to collect this phenotypic data is critical for this improvement in genetic improvement of estimating methane emissions.
Great, thank you, Lizzy. So the rumen is what really makes the cow unique. And Francisco highlighted well that this is one of the first projects that has so thoroughly encompassed the rumen microbiome into feed efficiency or methane emission projects. So Hilario, do you want to tell us a little bit about that component of the project?
Yeah, I'll be happy to, Heather. So like Francisco pointed out in the beginning of this presentation, the microbes that colonize the gastrointestinal tract of the ruminants, particularly the rumen, are the main responsibles for production of methane. And in order to evaluate this and compare like the differences between the animals, we need to actually, I think this is something that Francisco wanted to. Can you go ahead? Okay. Okay, we need to actually access the rumen microbiome. So the effort that we are doing right now is to actually sample the rumen microbiome, but also collect samples from other sites in the animals, so we can have potential phenotypic associations with the behavior of the animals in terms of methane production. So we have this group of high methane producing cows and the low producing methane cows. So we're trying to assess that. So in these pictures, you see Alice Perez, who's a PhD student in the Department of Animal Dairy Science that leading the effort together with Guillermo Bojo, a postdoc in Francisco's lab, in collecting these samples. So in the center of the slide, you see the stomach tubing technique being applied to collect rumen fluid from these cows. So we're phenotyping the animals, separating the animals from the high and low methane emitters. And this is one of the approaches they're using to collect the rumen fluid from the animals. And on the left side, you see Alice performing buccal swabbing, which just like had a talk about the buccal swabs and how we can use that to phenotype animals as well. And also on the right side, the collection of fecal materials from these animals as well. So we have like a direct access to the rumen environment, but also to other sites that could be used as predictive tools to associate with the other phenotypes that we're seeing in these animals. And the idea in this part of the project is to actually compare the microbial populations between these two different groups of animals and determine these differences in terms of community membership and all the aspects related to that. And so we can have like a better understanding, mechanistically speaking, how this process differs between these two animals. And one thing that I wanted to highlight, I think is the next slide, when you look at the cow itself does not produce methane. I mean, the microbes, especially a specific group of microbes in the rumen of these animals, are the ones that produce methane. So this group is the archaeal group. They're very specific type of metabolism, very specialized in terms of the substrates they utilize. So understanding these differences and the major groups that colonize these different types of animals, if there are differences, or learning through the process how this goes, we will be better suitable to actually use additives and actually modify this ecosystem and actually manipulate the methane production in these animals. So this is one of the main objectives in that part of the project. And like Francisco highlighted, in this part of the objective, we also have the rumen swap experiments that are going to exchange contents of animals with high and low methane emissions, so we can have like a better assessment of the host and the microbial community role in the process as well.
Great. So all joking aside, it really does take all aspects of the collaboration to make a project of this scale and multidisciplinary nature work, right? So Francisco, can you summarize for us the impact that this has had over the last few years? What progress are we making? Are we making any progress? And how do we quantify it?
Good question. Let me go back. Okay, so first of all, as a group, and I'm talking about the fall and grant universities, the relationship with the USDA and the Council on Dairy Cattle Breeding. This relationship started in 2010, 14, 15 years ago, when we started to work in dairy cow feed efficiency. As a result of that, in December 2020, we implement a national genetic evaluation for dairy cow feed efficiency. This idea of if I'm having two cows that produce exactly the same amount of milk and they have the same body weight, but one is eating less than the other one, that one is more profitable, right? Feed efficiency has also a positive impact on the environment. So we are exactly the same group that developed that trait, and now what we want to do as a natural next step is to develop a national genetic evaluation for methane emission traits. Now, for those that are not geneticists, let me sell you the tool of genetics. And for that, I'm having here real data. This is the change in milk production per cow per lactation in the last 60 years, right? It's easy to see that the dairy cow today, she produces more than twice as much milk as a cow of 60 years ago. And 60% of that change is due to genetic selection. In fact, genetic selection is a very powerful tool for achieving lasting gains in dairy cattle performance. And contrary to changes or improvement that we can achieve due to changes in management, nutrition, and cow comfort, the changes that we achieve through genetic selection are permanent, cumulative, and incremental, what makes genetic selection a very powerful tool and very cost-effective strategy. So we talked about feed efficiency before, and this is a picture of the population right now. This is in Holstein. We are using extreme cows here, the most efficient one and the less efficient one. And you can see that regardless the production level of those cows, if we are talking about 30 kilos, 40 kilos, 50 kilos, or 60 kilos, there is a beautiful difference between the amount of feed that those cows eat between the less efficient and the most efficient. We are talking about 10, 15 kilos of dry matter intake per day. Again, it's an extremely powerful tool. This is one of our recent trials. In pen 30 is the pen that Lizzy showed that we have in Arlington, where we have 32 gates and we have 2 green feeds. So we can phenotype there 64 cows. So each box plot here is a cow. And we evaluate the cows between 7 and 8 weeks. And as you can see here, this group of cows, we are talking about multiparous cows, they produce roughly 450 grams of methane per day. What is important is the variation. There is a beautiful variability. You have some cows in the same pen under the same diet that they produce roughly 600 grams of methane per day. And you have other cows exactly in the same pen under the same conditions that produce roughly 300 grams. So there is a beautiful variability in methane emissions in dairy cattle. And now we know, based on our preliminary result, that roughly 25% of that variation is due to genetic factors. So what we plan to do is to implement a national genetic evaluation so we can start to select and breed those cows that will reduce methane in next generations.
Exciting stuff. All right, questions from you all. I certainly have a list, but I want to make sure we have time to take questions from the audience. Yes, Jimena.
Thank you for the nice summary. I have a question. I think it's for all of you. You were saying that selecting the cow, like you're selecting the cow genome, but then Hilario is saying that methane emissions obviously arise from the bugs in the rumen. When you select a cow, do you indirectly select the rumen microbiota that she's going to have for life, or boom?
It's an excellent question, Jimena. Yeah, no, there are data out there. I mean, we like many years ago, we did a study, the time that I was actually visiting UW-Madison with Paul Weimer, we did a rumen swap study. I think was the first rumen swap study that was done at the time, to evaluate exactly the contribution of the host and the microbiome associated with the host. And one thing that you see, and that study was repeated after, a few years after, involving Paul Weimer, Garrett Soon, and other researchers at UW-Madison with high and low feed efficient animals as well, and to look at the control of the host over the microbiome. And there is directly like an interaction between the host and the microbiome. If you swap the contents of the animals, they tend to go back in terms of functionality. So VFA production, several aspects of the community membership in terms of composition returns to the previous state before you actually do that. So there is a major component of the host that determines the microbial community that's associated with that particular animal. And I think this side that Francisco was talking about, the association between the genotype of the animal and the microbial community associated with that, that's going to be an interesting aspect of this project that we can tackle, I think, with the results of the microbiome sequencing and look at the membership of these animals as well.
Thank you. My question is about clarification of what we really mean by emission. And here's the way I'm going to phrase the question. So 10 years from now, when this is all said and done and we can apply the result into the industry, are we going to have less methane emission per cow? So is methane production going to be reduced? Or are we going to have less methane production per kilos of milk, methane intensity going to be reduced? Or are we going to have both? Or having one is contradictory to the other? That's the question I have for you.
Yeah.
Thanks.
Thank you, great question. So, so we are measuring methane production, right? Amount of grams per day. As you can imagine, cows that tend to eat more, they tend to produce more milk. They also tend to produce more methane. So if you target methane production per se, well, you are looking for troubles, right? Because you will have a negative impact on production. So we are working on what we call residual methane. It's similar to residual feed intake, that is methane production adjusted for production and metabolic body weight, and we call that residual methane intensity. That is one trait. And that is a trait that we can measure on research farms, because in order to measure that trait, you just need methane production, milk production, milk composition, and body weight. And we can also measure that trait on commercial farms. And also we have methane production adjusted for dry matter intake that we call residual methane yield. And that's a trait that so far we can only measure on research farms where we also collect feed intake. So the good news is that those two traits are highly correlated, as you expect, because the two predictors, milk production, metabolic body weight, and dry matter intake, are highly correlated. So you end up with similar residuals. And we are talking about a correlation around 95%. What is very good, because that gives us the opportunity to go to commercial farms and start to collect data and enrich the database that we are generating in research farms. So those are great news. So going back to Michelle's question, in the future, we will need to include methane emission trait in a selection index, because at the end of the day, our farmers, they do not select for a single trait. They select for multiple traits at the same time using economic selection index, and that index will probably have methane production and also these residual methane traits.
I guess I just wanted to also add, I think, I mean, even outside of this project and as in our respective areas, it's also going to be important to estimate the volatile solids aspect from manure as well. And if we are improving some of the digestibility, what does that mean inherently from the emissions from that aspect as well? Because we're obviously only talking about enteric.
Great, Marta had a question in the back.
Hi, I find so interesting to look at this graph that shows the variability, because as we know, if there is variability, there is opportunity for selection, right? I'm wondering if all these animals were on the same diet, right? If there is an opportunity to select animals for specific diets. So, for example, in regions of the world where there is not as much grain or silage production and there is more grazing as part of the dairy industry, if there is opportunity to select for divergent populations, so populations that will select that will be more efficient or less methane producing when they're grazing compared to on a grain diet, or if it is expected that those traits will be different enough that they will allow for different populations.
So, great question. So, for the 64 cows that I show, they are exactly in the same diet. So that's real variability under exactly the same conditions. Now, your question is fantastic, and it's probably what we will try to address in the near future. That is basically the genotype by diet interaction. So basically is something like this. if we have a bunch of cows under this diet, let's say a diet rich in corn, and we rank the cows from the cow that has the most emission versus the least emission. We rank the cows. The same group of cows we put now in a diet very rich in forage. If we observe a re-ranking of the animals, then we are in troubles because basically there is a genotype by diet interaction. Now, our selection tools will depend on the diet that we are addressing. So the idea to work in genotype by diet interaction it's in the pipeline, and we will try to address within our system, right? It could be a diet very, very rich in corn or in forage. We are not probably addressing grazing, that is the other extreme, right? It's within our diet, but with some variation, try to address if there is actually a genotype by diet interaction. Thank you. That guy knows more than us, so I don't know who will answer this question.
Thank you for the presentation. I have a maybe more general question. This you are starting this project, but how do you imagine in maybe 3 or 4 years? I hope that we have the 4,000 cows phenotyping, but for the future, because nowadays we have a genetic evaluation for residual feed intake, but one of the bottlenecks is how to collect this data in commercial farms. So how do you imagine that we can continue with methane emissions in all across US? That is for maybe the part of phenotyping and how to implement the genetic evaluation, and if could be some synergy between methane and residual feed intake. And the other question, maybe is more for Hilario, is about the rumen microbiome and how you imagine if it is more, not maybe more for the genetic part, but maybe use the microbiome for intervention with additives, and if the idea is to test some additives and how to use it. Thank you.
I go first. No, that's a great question. I think we have an advantage compared with feed efficiency. So I told you, right, we started to work in dairy cow feed efficiency in 2009, 2010. And until today, the phenotyping only occurs in research farms, in the 4 land-grant universities that I mentioned and the USDA. And we have right now a reference population of 10,000, 11,000 Holstein cows. And with that, we actually implement a national genomic evaluation using genomics, and it works. Now, the advantage that we have with the methane emissions is that we can measure methane production in commercial farms using, for example, the green feed. And this is what we are doing right now. So in short, I see in a few years, I think we will have more and more green feeds in commercial farms, or at least in some farms where we will do the deep phenotyping. The other question is, and those are Guillermo's results, is our preliminary results are showing that the more efficient cows tend to produce less methane. And this is a win-win. These are great news. That means we have been reducing methane emissions since the implementation of feed efficiency. And if we start to select directly for methane emissions, then we will be moving in the same direction, indirect and direct, reducing methane emissions.
And along those lines, so we're continuing to, again, make more measurements on commercial farms as well at USDA, and particularly in younger animals. And also that some of the newer technology coming out, for instance, boluses that are measuring methane on individual animals. So looking ahead, I think we're going to have some other technologies that we're comparing to some of the standards, such as the green feed, and be able to implement that on more commercial farms and get direct measurements.
As relates to the microbiome, Jeremy, there are many aspects of the difference between the animals and the ranking of the animals associated with the composition of the archaeal population specifically they were looking at, but also potential microbes that produce the hydrogen that are actually used by these microbes, the archaeal population, to generate methane. So understanding like the specific population associated with different types of animals ranked in the different groups, and the substrate that they use as a preferential like substrate for methane production can lead us to develop more like targeted approaches in terms of additives to specifically look at this, like the effect on these microbes. There is one aspect that is also related to the diet effect that Francisco mentioned that I think would be really valuable to associate that with the level of the population of these specific archaeal groups. And how that, because if you change diet, you also expect to have sometimes like effects on passage rate, for example, and that could have a like, huge impact on the activity of these microbes as well. So these are factors that are not like accounting right now, but in the future, it should be also taken into account for modulating of the microbial population there.
All right, so I'll jump in with a question here, and then I see we have one over here. So first, you mentioned diet, and I think a lot of times we think about a diet that might reduce methane, but something that I think a lot about is, what if it's something that we're feeding the cow that she's using to produce feed for humans, so she's producing a human consumption food product that would otherwise have been a complete waste. So not even thinking beyond corn stalks, which I don't think any of us enjoyed for lunch, right? Although all of our cows did. But I'm thinking about waste products that would go to a landfill and as they decomposed would create methane. And instead the cow is consuming that and creating milk and generating methane, but that methane might introduce in neutrality. So for example, Dave Daniels, the chair of our advisory council, was just telling me this morning, and I hear this often, but you were just telling me that you're going to feed cookie waste on your farm starting later this week, right? So broken cookies that would otherwise go to a landfill, and now cows are using those nutrients to make something that is for human consumption. So my question is, how do we incorporate that into the way we tell a methane reduction story? Because even if that methane production is 1-for-1, there's a methane and a waste component that didn't happen in a landfill, right? So how does that become part of the story?
On the microbial community side, I mean, that's something that we can actually test and to see how that impacts the composition of the microbiome and also the levels of these microbes associated with methane production also, and the impact on the overall methane production. If you feed that to an animal and reproduce the effect, that's something that you can actually use to modulate the effect on methane production. That's actually one of the things that people have been trying. Different types of feeds, different types of supplements in the feed, like tannins, like fatty acids, and other things. But if you use these locally sourced products to actually feed the animals and have an impact in reducing methane, that's really a powerful thing because it has an economic impact, actually. So that's something that could be evaluated.
Yeah, it seems that it needs to come down to the whole life cycle analyses of what we are evaluating, kind of speaking to what Hilario was mentioning, and looking at how it fits into the bigger context, is what you're saying, waste streams and what it would otherwise be used for.
Great. Question over here from Mike.
Yeah, can you comment on how the milk sampling piece of this is going to play in the future and what you envision that will gain us as an industry?
So, so what we are, when we do milk testing, right? When we do milk testing, it's basically to know the composition of the milk. And for that, we use spectroscopy. And basically what we are thinking of is try to use the output of that machine, use the very raw numbers from that machine and develop a predictive model that we are able to somehow predict methane production per cow. If we can do that, and our preliminary results are showing that we are able to predict that with a quite nice accuracy, we will be able to every time that a farmer sends samples to a milk lab and get back, fat, protein, lactose, somatic cell score, we will also get a prediction of methane emissions. And eventually, if we need to minimize or mitigate methane emissions using, for example, the use of some feed additives, we can target what are the cows that are emitting more in the farm and use those feed additives that we know they work, but they are extremely expensive only for that subset of cows that are emitting quite a lot, for example. We do not have tools today to actually identify what are the cows that are emitting more in the farm.
Other questions for our panelists?
Hello. I have a question about like clarifying some my understanding of things. So I guess the methane emissions get recycled in 12 years, and the number of cow heads in the U.S. have stayed constant over 20 years. And I guess the efficiency of cows are also increasing with years. What are other ways that what are some ways to explain that phenomena? Why is that happening?
So the idea is why cows are becoming more efficient, and at the same time we have the same number of cows, right?
What is adding on the more methane? Like methane emissions are increasing over the years, but the number of cow heads have remained constant over the years. So what is the reason for that?
So let me try to answer. So if you think about a cow, and Michelle probably can help me. If you think about the individual cow, the cow today is producing much more methane in terms of grams per day than a cow of 60 years ago, because it's a cow that is eating much more than a cow of 60 years ago, right? So if you think about methane production per cow, maybe it's going up. Good. If now you adjust methane production for milk production, maybe it's going down, because the cow is producing more methane, but she's producing much more milk than 60 years ago. So when you play that efficiency, that ratio is going down, right? Now, if you want to target methane emissions today, we don't have the tools. So we don't have genetic tools to directly select and breed for reducing methane emissions. And we don't have the tools to go to a farm and say, that cow, she's producing more than the mean, and that cow, she's producing less than the mean. We don't have the tools. And I think part of our project is actually, the goal is developing those tools, either for selective breeding, either for pure prediction and management, and at the level of the microbiome, even to develop proxy traits that we can identify who are those cows.
Other questions for the panelists? So I have one, I'll jump in here while people are thinking. We do still have a few minutes left, so if you have a question you're brewing, please go ahead and form it. But this seems to be a pretty abstract trait as we're talking about residual feed intake, residual methane emissions, methane intensities. It's fairly relative within a set of cows. So is there a very tangible trait that we're using with farmers, and how do we explain this to our end user who's going to implement this? Because even if we make progress within this set, what we want to do is make genetic progress in the global herd, right? So how are we explaining this to farmers in a way that's really tangible?
Well, as I think about it, I mean, in general, we're actually improving our nutrient use utilization and feed efficiency as we typically look. I mean, what we're looking at now and selecting these animals that may be consuming less feed, still producing greater amounts of milk, and we know we're reducing their emissions. So I think as we look for it's to make it tangible, it's being able to select animals that, again, as you look at your income over feed costs, are going to be the ones that are creating more income for their feed costs and more efficiency. I guess as I look at just the general simplicity as far as like intake versus emissions.
So one way to explain this residual methane trait, right, is imagine you are in front of two cows. They are producing exactly the same amount of milk, both, and they have exactly the same body weight, so they have the same metabolic body weight, but one cow is emitting less methane than the other one, right? That cow has a negative residual methane while the other one has probably a positive. You can think the same when you think about the residual methane yield, that is this idea of adjust methane by intake. You are in front of two Holstein cows, they are eating exactly the same dry matter intake, but one is producing, 100 grams of methane less than the other one. That one is probably, or without probably, more environmentally friendly, right? But it's also more efficient because going back to the beginning, you can think of methane also as a energy sink, as a loss of energy between 6 and 12% of gross energy that could be used, for example, for milk production. So that will be one way. Now, how we implement, probably we end up like, feed safe, this idea to transform a negative scale in a positive scale, something that could be methane safe. At the end of the day, Heather, the key is to introduce those methane emission traits in economic selection index. And for that, we still have a gap, huge challenge, that is find what is the economic value of methane emission traits. We can think about that in a positive way. That means the industry will stay to pay more for milk that comes from cows that produce less methane. That is one positive way to think about it. Or maybe the federal government, we will receive some benefits if our cows produce less methane. You can think about it in a negative aspect when more what is going on in Europe now with dairy farming and the environment, you have to do it or you are out of the system. In both the scenarios, you can come up with economic value for reducing methane, and that will be the way to implement those traits in an index.
Thank you. All right, any last questions for our panelists? Yeah, go ahead, Jessica. There's a microphone right behind you.
So I will maybe conclude us with a big picture question. So when I think of this, most of us in this room have talked about methane emissions. We've talked about sustainability on farms. I interact with a lot of people in the public sector who have a lot of misconceptions on what sustainability in the dairy industry means and what methane emissions actually do. One of those biggest misconceptions, right, is when people think farms, they think about the dairies, they think about the manure and the effects that that has on us. And I think it's very common that a lot of people believe that the highest level of methane emission is actually coming out of the back end of the cow and not the front end, like we all know it to be. So I think a lot about what we've seen today across all of the sectors and the conversations we've had, from soil science all the way to here. How does the Dairy Innovation Hub wrap all this together from a sustainability standpoint, not only from the emissions out of the front end of the cow, but what happens on the back end of the cow and how that, wraps into, the soil science and the forage aspect, the whole circle of stewardship and how we actually get there?
Yeah, that's a great question. You're all pointing at me.
Yeah.
So I do think this is a great one to end on. So from my perspective, the Dairy Innovation Hub is here to help prime a lot of these things, right? We've been a part of funding or co-funding a lot of the efforts that you've seen presented here today and many of the ones that you mentioned. But the other thing we do, which actually ties to what my final thoughts were for today, is look at everybody who was in this room today. We had 200 people from campus who are all focused on dairy. And we're all here together talking about what we have and what our stakes in the dairy community are. And those are conversations that didn't happen a few years ago because we didn't have reasons to gather together. And collaborations tended to be smaller and more discipline focused. As our science has become more multidisciplinary, we need these opportunities to gather together and to talk about these bigger systems-wide, which we heard Lizzy reference earlier, approaches, right? So even right now, we know that there are ideas, proposals being worked on. There are NSF projects on sustainability from the human side that are encompassing everything from water management, nutrient management, all the way to the cow herself and to food systems. So I think from the hub's perspective, it's our role to be a part of those conversations to help connect people who can take the leadership on those. Most of those efforts that you're talking about that tie everything together are going to be much bigger than those of us in the room. They're going to be bigger than any one discipline. They're bigger than cow biology, which was a vision that we've had since the inception of the Dairy Innovation Hub. They're encompassing of many different disciplines and are going to take a lot of people and a lot of universities and research stations to tackle. So one of the things that I've been so excited to see now in year 4, we're at maybe 4.5, is that we have been able to be the catalyst on some of those really big conversations that are happening. And I think that that's a very exciting time for all of us, right? So I think it's bigger than just the hub. I think it's bigger than any one of us as a researcher, because it's definitely a systems approach to package it all together, but it's something that's needed. I sat in recently on a carbon credit, carbon accounting workshop that PDPW put on, and it was the same thing where industry partners are saying just that too. They're saying, okay, we can do this one part of the calculation, but how do we take that math and how do we do that across everything? How do we incorporate the economics? How do we incorporate the farm system, the consumer side, the trucking side? All of those aspects have to come in. So I think it's a great question. Anybody want to add anything?
Just a quick comment, because I think Jessica touched on a point that's really, really important, like looking at the farm like holistically. And I think this morning we had a great presentation on that. Dr. Zubacher actually touched on the manure problem, and this is something that we definitely need to expand research on. As she pointed out, 30% of methane in the farm level can be produced like from manure. So even if you tackle, like if you look at the most like effective additives that are used for cattle, that's pretty much like the range that you're looking for methane reduction, right? Between 15, 30% of reduction. This is inside the animal. So it depends on the animal eating the diet with the additive and modifying the ecosystem to reduce methane production. With manure, it's outside the animal. There are other approaches that you can add directly there to actually reduce those emissions that the farm level are also very important. So you have to look at the whole ecosystem, I think.
Great, and thanks for that great big picture question. With that, we'll bring this to a close. So I do have some final comments, but before that, let's thank our panelists. All right, so as I mentioned, one of the most exciting parts about the symposium and something that you all gave us feedback on last year and the year prior was just the opportunity to gather for the day, for everybody to get together, to network, to build collaborations, to talk about dairy. In my 11 years here at UW-Madison, before the Dairy Innovation Hub, there was only one day like that, and it was awkwardly like a speed dating. Everyone talked for 30 seconds about what they did in dairy. We hope that you find this to be a great opportunity to network, to collaborate, to hear some of the other things going on across our 3 campuses, and as an opportunity to follow up on those people you've met today or heard. So with that, we want to transition to an opportunity, a final opportunity to do just that. We do now have a reception. There's appetizers. I've been told it's heavy appetizers, which means Maria picked the great things on the menu. So we will transition to that and have posters. There's a few things I want to note. There are flash talks in the back. These are students that are competing for a $1,000 no restriction travel scholarship to go present their research. Your blue ticket and your name tag, after you listen to those flash talks, can be put into the buckets that have their names on them. That's how we'll be voting. You also have drink tickets. We have beverages in the back, and the balcony is open, and it's gorgeous out there. A final thing I want to say is that these events are not possible without a great team. So I'd like to give a hand to Maria Wolt, to Elizabeth Burnt, to Jory Selensky, and some folks from Cal's External Relations who came to help. So let's thank them, please. They did fantastic. Thank all of you for coming. I know this is a busy time of year. We intentionally put it right after finals, so you're not teaching, right before the end of the 9-month appointment, so you might still be here. But asking a whole day of your time is always a huge ask, and we so appreciate you being here with us. So with that, thank you very much for coming, and we hope you're already planning to come to the summit in November. Thank you.